Hello, welcome to another lecture. This is chapter 5 lecture, and we're going to be discussing membranes. So we know within these membranes, they are structures found within cells, and they are phospholipids arranged in a bilayer. They are globular proteins that are inserted into the lipid bilayer, and we can see these in the form of the fluid mosaic model, where the mosaic of proteins floats in or on the fluid lipid bilayer that like boats on a pond. And you can see that in this image here where we have the different types of the membrane transport from those different proteins embedded in that membrane. Now remember that membrane is composed of a polar and non-polar aspect. So we have the hydrophilic, which is polar, and these are polar heads of the phospholipid bilayer. We also have the hydrophobic, which is the non-polar fatty acid tail of the phospholipid bilayer. So we have a hydrophilic on the outside and the hydrophobic on the inside. This allows for the movement of water, right? So remember, hydrophobic is meaning that it's water-fearing, and hydrophilic is water-loving. We have simple diffusion that goes on with molecules going straight through the membrane. We also need these different transmembrane proteins to allow for this movement. Now, the structures of phospholipids can be shown here, where we have the nonpolar hydrophilic tail in this chain of molecular compounds, along with the polar hydrophilic heads at the top here. There's a fluid mosaic model of the cell membrane shown in this image that lets us see deeper into the cell in the different bilayer that we see along this membrane. There are extracellular matrix proteins along with glycoproteins that are involved in cell function. Cellular membranes have 4 components. We know that these 4 components include phospholipid bilayer, the transmembrane proteins, interior protein network, and cell surface markers. Within the phospholipid bilayer, we can say that these are the flexible matrix. This is the flexible matrix of the cell, and which allows for the barrier of different objects to come in and out of the cell. So it's a barrier to permeability. The transmembrane proteins themselves are integral membrane proteins, which we saw before, that are embedded in the membrane of the phospholipid bilayer. And the interior protein network is where we find the peripheral or intercellular membrane proteins. Lastly, we have these cell surface markers that are the glycoproteins and the glycolipids that make up these cell surface markers themselves. Right? So it is important for us to understand these different receptors and the plasma membrane and all the different components that accompany cell permeability. So studying the cell membrane allows us to see further by looking at the transmission electron microscope, TEM, and the scanning electron microscope, the SEM, that are used to study membranes. One TEM method is to embed the specimen in epoxy. So you are able to see further into this organism. Now, this is greater than 1 micrometer thick shavings of the embedded specimen that can be imaged. Shown below, we can see cell 1, cell 2 here. And within this, the plasma membrane of cell 1 on one end and the plasma membrane of cell 2 on the other. And from there, the transition and the embedded space between is the cell junctions that allow for movements to occur. So we see that there's freeze fracture and microscopy that visualizes inside this membrane. As you can see, we take a cell that is frozen in medium, and it is cracked with the knife here. Now that knife is allowing us to cut and remove part of the cell as it fractures that interior hydrophobic area of the lipid bilayer, splitting the plasma membrane into the 2 layers shown here. Now, the plasma membrane separates such that the proteins and other embedded membrane structures remain within one or the other layers of the membrane. So we see here the fractured upper half of the lipid bilayer that is exposing the lower half of this lipid bilayer. And as we continue to form, the exposed membrane is coated with the platinum, which forms a replica of the membrane. Now that platinum is important, right? Because that replica is needed for the underlying membrane and its understanding and how it's dissolved away. And the replica is then viewed with the electron microscope where those photons are hitting the outside of that organism for us to be able to see. All right. So we also now have our phospholipids and the structures that consist of the glycerol, a 3-carbon polyalcohol, along with the polyalcohol, along with the two fatty acids attached to the glycerol. And these are nonpolar and hydrophobic. We see that these are also water-bearing. Phosphate groups are attached to the glycerol, and polar and hydrophilic means water-loving once again. So spontaneously, these are forming a bilayer. This bilayer is hydrocarbon tails that are inside and polar heads that are grouped outside outwards. Phospholipid bilayer also has fluids, right? So the bilayers are fluid, and hydrogen bonding of water to itself and to polar heads holds the layers together. We know that individual phospholipids and unanchored proteins can move through the membrane, allowing for different solutes to move back and forth through cells. And the same can be said when we're talking about the fusion of cells from human cells to mouse cells, where there's a fusion of the membranes that can intermix those proteins that are on that membrane. Now, this understanding of the phospholipid helps us go forward into the mechanism of fluidity. So the influence on fluidity occurs through the understanding of saturated and unsaturated fatty acids. Saturated fatty acids make the membrane less fluid than unsaturated fatty acids. And we see that if we're making something less fluid, then we're going to be more solid, right? So the saturated form of those acids are actually more solid than the unsaturated fatty acids. So warm temperature makes the membrane more fluid than cold temperature, and cold tolerance is in bacteria is due to the fatty acids desaturizes as it denatures. Now, lipid composition of the ER membrane allows for Golgi stacks and plasma membranes that are distinct within this membrane. So they're composed of the effects of fluidity, thickness, shape, and the thickness shape of the membrane, in which are formed based on the saturated and unsaturated fatty acids. Once again, the saturated has the straight form, as the unsaturated has a kink in the tail here with a double bond, which shows that it's going to take more energy to break this, right? Because there's a double bond compared to a single bond. So when we're thinking about fatty acids, the saturated ones are technically more healthier, right? Because they're faster to break down, easier to break down in regards to this bond shown here. Now we have membrane proteins that include various functions that have transporters, enzymes, cell surface receptors, cell surface identity markers, cell-to-cell adhesion proteins, along with attachments to the cytoskeleton. And all this happens within the membrane proteins. You can see on the right that image that shows the bilayer and that membrane, the single transmembrane proteins that are there, along with the multi-pass proteins, peripheral membrane proteins, cell membrane itself, along with that lipid anchored protein. Now, all of these things allow for proteins to function, right? So the transporter allows for molecules outside the cell, inside the cell, enzyme components where we know that there is a receptor for protein that is going to allow different analogs to connect and bind. We also have cell surface receptors that after binding send off signals and chemical signaling to the remainder of the cell. Also, cell surface identity markers that are marking and identifying different aspects of other cells and cell membranes for future information. We have cell-to-cell adhesion as well, where different proteins in that membrane are connecting, allowing for adhesion amongst and between different cells. So cells are communicating as well. Attachment to the cytoskeleton also occurs on the membrane using these different proteins, along with the fibers that we have talked about previously. These structures are related to function. So we know there's diverse functions arising from the diverse structures of membrane proteins. They have a common structural feature in which they are related to their role as membrane proteins. Now, peripheral proteins are anchoring molecules that attach membrane proteins to the surface. As we see in this image, here's the peripheral protein that is loosely bound to the surface of the membrane that we just defined. Within anchoring molecules are modified lipids with nonpolar regions that insert into the internal portion of the lipid bilayer. We know the chemical bonding domains that link directly to these proteins are the second part of how these anchoring molecules occur. So we can also see this in the form of anchoring junctions, where the They attach the cytoskeleton of a cell to the matrix surrounding the cell or to the cytoskeleton of an adjacent cell. So we can see here that the cell 1, cell 2, along with those plasma membranes are found in this image. And in the middle, we have the intercellular space. Membrane proteins are integral membrane proteins that span the lipid bilayer. These are the transmembrane proteins. Non-polar regions of the proteins are embedded in the interior of the bilayer, and we know that polar regions of the protein protrude from both sides of the bilayer. So transmembrane domains allow for that spanning of that lipid bilayer and the hydrophobic amino acids that are arranged in the form of a alpha helices. Now, the alpha helix and the understanding of this transmembrane domain can be shown here, where we're able to see these different proteins in their form and how things are evolving, moving for macromolecule transport. These transmembrane domains shown below are proteins that need only a single transmembrane domain to be anchored in the membrane, but they often have more than one such domain. So we have here one domain along with the multiple shown here that allows for G-coupled protein reactions using the different ATP to open and activate some of these proteins. So we have proteins that need to be activated, but we also have these pores, these nuclear pores that are extensive nonpolar regions within a transmembrane protein that can create a pore through the membrane. The cylinder of the beta sheets in the protein secondary structure are called a beta barrel, and the interior is polar and allows water and small polar molecules to pass through the membrane. As you can see in this image, here's the membrane, and the pore is pretty obvious how open that pore is, right? We also have the nuclear envelope that is this area of the membrane. Within passive transport is the movement of molecules through the membrane in which no energy is required. Molecules move in response to a concentration gradient, and we know that diffusion is the movement of molecules from high concentration to low concentration. And this will continue until concentration is the same in our regions. Remember, this form of being the same in our regions is also back to our term homeostasis, in which all organisms and all animals are looking to go back to this form of in the state of homeostasis. Regardless of what spectrum or what end they are on, homeostasis is always the objective. Now, passive transport allows for the movement of particles with no energy, and molecules flow across that membrane by diffusion. The active transport allows and needs required energy in the form of ATP or NADPH to allow these molecules to be pumped across the membrane. And like I said, ATP is required for active transport. Now, when it comes to diffusion, we know that diffusion is like dropping a food coloring or this blood drop or something like that into a beaker of water or into a solution that allows for it to diffuse in that medium. Now, diffusion is where we see the particles separate and spread out amongst the container and the fluid in which it is in. The transport across membranes is important because a major barrier to crossing a biological membrane is the hydrophobic interior that repels polar molecules, but not nonpolar molecules. Remember that nonpolar molecules will move until the concentration is equal on both sides and is limited to permeability to small polar molecules and is very limited permeability to larger polar molecules and ions. These being shown below. An example being large uncharged polar molecules such as glucose and sucrose that we talk about all the time are not able to permanently just be sent across the membrane. So they would have to be put into different forms of hydrophobic molecules like oxygen, carbon, nitrogen, and small uncharged molecules like H2O, glycerol, urea, and ethanol go straight through the membrane there. The others need forms of channels to help that happen. So we also know that there's facilitated diffusion, where molecules that cannot cross the membrane easily may move through the proteins that I just talked about, and they move from higher to lower concentration. The channel proteins are hydrophilic channels when open, and the carrier proteins bind specifically to molecules that they assist. So membrane is selectively permeable when it comes to these facilitated diffusing protein membrane spaces. We also have channel proteins that are ion channels. They allow for the passage of ions through the nonpolar interior of the plasma membrane, and the gated channels that open or close in response to stimulus. Now, these are chemical or electrical, right, based off of what needs to go on within the cell and the specifics. But there are three conditions that determine direction in which this happens. So relative concentration on either side of the membrane, remember homeostasis, and diffusion, along with osmosis, are things that are playing a role in how molecules move. So there's also voltage differences across the membrane, along with gated channels, and the channel is either open or closed. In this image, we have the top view of these channels that either open or close based off of the messenger and the receptors or the chemicals that are involved in the diffusing process.